So now we've spent a lot of time talking about the nutrients that are in microbes and how we can categorize those microbes based off of their energy sources and their carbon sources. Let's talk about how they eat, how they physically get the stuff they need to stay alive into their cytoplasm, how that stuff crosses their cell membrane and their cell walls. These transport mechanisms are very essential for microbial life. We need to get those necessary nutrients across the cell membrane and, if it's bacterial or fungal organism, across the cell wall as well. The main driving force of all of this is going to be the molecular movement and atomic movement. And we could go down a huge tangent and rabbit trail on thermodynamics, which we are absolutely not going to touch with a 10-foot pole because this is not a class on thermodynamics. This is a class on microbiology. So first, let's talk about diffusion. Diffusion is this phenomenon that's been observed in nature. It's just molecular movement. Things generally spread out from areas of high concentration to areas of low concentration. This is also known to be powered by the law of entropy. A good example of this would be if you took perfume and sprayed it in the air. There would initially be a really high concentration of those perfume molecules in the air immediately where you sprayed it, but eventually they would spread out and fill the room with an even concentration of perfume molecules and an even concentration of that smell, whatever that smell may be from the perfume. So for diffusion, we will always have areas go from an area of high concentration to an area of low concentration. Diffusion does not require the input of energy. It happens passively, all by itself. Osmosis is a very special kind of diffusion. It's the diffusion of water through a semi-permeable or selectively permeable or differentially permeable membrane. However you want to skin that cat, that membrane will let some stuff through and not other stuff through. One of the things that is let through the membrane is water. And this water is going to move in such a way that it'll go from an area where there's a higher concentration of water to a lower concentration of water. Some membranes will be designed in such a way that water cannot go directly through them and has to go through special channels as opposed to the main body of the membrane. These special channels that allow water through are typically called aquaporins or water holes. Sometimes these membranes are going to be designed in such a way that water can travel freely through the membrane regardless of the aquaporins, but larger ionic compounds or even larger macronutrients or macromolecules cannot move through the membrane. If our membrane is designed in such a way that there's a liquid with a high salt concentration on one side and a low salt concentration on the other side, and water can travel through the membrane, the water will move through the membrane in such a way to equalize the salt concentrations. So we could have, for example, pure deionized distilled water on the outside of our, what's the term, dialysis tubing right here that we have in this beaker. And then on the inside of the dialysis tubing, we could have a bunch of sugar water. And essentially, that's just a bunch of red molecules I'm arbitrarily calling sugar, and then blue molecules, which I'm arbitrarily calling water. And since there's a high concentration of water on the outside, it's 100% water on the outside, and it's less than 100% water on the inside, water is selectively going to move into this tube, this dialysis tubing, and the dialysis tubing will swell up. This is the basis of the classic high school biology experiment where you take eggs that have had their shells dissolved off, and you take one egg and you put it in corn syrup, you take the other egg and you put it in distilled water. One egg swells up, the other egg gets smaller, and then you record the changes in mass based off of the movement of water with its concentration gradients. Now, if we looked at a bacterial cell, the cell wall of that bacterial cell is going to keep the bacterial cell from exploding. If we're looking at a protozoa cell that doesn't have a cell wall, that protozoa cell only has a cell membrane to give it integrity, to keep its inside separated from the outside. And that protozoa cell may be subject to explosion or cellulitis. So let's look at an isotonic solution. Iso meaning the same. So in an isotonic solution, the concentration of our salts and water are equal in and out of the cells. So there's no net movement of water into or out of the cell. So the cell is going to be nice and happy. It's going to stay the same size and stay the same mass. In a hypotonic solution, there's a much higher concentration of water outside of the cell and a higher concentration of salt inside of the cell. And in those situations, those hypotonic solutions, there's a net movement of water into the cell. And as that water moves into the cell is going to swell and increase in mass. Now, if it's a bacterial cell with a cell wall or a fungal cell with a cell wall, the cell wall is going to keep it from bursting. If it's a protozoal cell or another cell type that doesn't have a cell wall that's put in that hypotonic solution, the cell is at risk of lysing or exploding. And then finally, we have hypertonic solutions. In hypertonic solutions, there's a higher concentration of water in the cell and a higher concentration of salt outside of the cell, or dissolved solute outside of the cell. So there's going to be a net movement of water out of the cell, and that net movement of water out of the cell causes the cell to shrivel up and reduce in mass. Now, if there is no cell wall, the cell is going to become visibly smaller as it loses water. If there is a cell wall, the cell wall will remain the same size, but the cell membrane within the cell wall is going to shrivel up for those organisms that have the cell walls. Let's review our forms of transport processes. We'll talk about the description on this PowerPoint slides first, and then we'll come back to this figure. So we have active transport. Active transport, by definition, requires energy, the expenditure of energy. It's always going to move something against its concentration gradient, or against the direction of diffusion. It can occasionally move something in the same direction as its concentration gradient, but if it's doing this and it's active transport, it has to be done faster, or it has to be assisted and go faster than how it would with normal diffusion all by itself. That's going to be the exception. Generally speaking, in almost every situation, active transport is against the concentration gradient, from an area of low concentration to an area of high concentration, so opposite of diffusion. Oftentimes, active transport is going to be powered by specific membrane pumps or proteins, and these pumps or proteins are going to typically require ATP to power them. So, if we look at monosaccharides, amino acids, organic acids, phosphates, or metal ions, these are all examples of chemical compounds that we're going to transport actively against their concentration gradients. If we have endocytosis, we're going to take something into the cell. There are two kinds of endocytosis. So endocytosis is a big umbrella term. We have phagocytosis. During phagocytosis, we're going to have the cell membrane wrap around something that's solid, or an entire cell. So when we have phagocytosis, which literally means cell eating, phago meaning to eat, we are going to have the cell wrap its plasma membrane around a molecule, a big, or a cell, or a big substance, and bring it inside as a vacuole. During penocytosis, which means cell drinking, the cell is going to wrap its plasma membrane, or endocytize some liquids, a small vacuole of liquid, and bring that into the cell. Oh. And I don't have an excellent, good figure for you for penocytosis. Let's look at our summary figure here for active and passive transports. We have simple diffusion. Simple diffusion, generally speaking, is going to be a substance just moving in the direction of its concentration gradient. Or we can have facilitated diffusion. Facilitated diffusion is when we have a protein carrier that carries that substance with its concentration gradient through a membrane that it would normally not be able to travel through. So there's a carrier that helps it move across that semi-permeable membrane. Both simple and facilitated diffusion do not require the input of energy. Active transport, by definition, requires the input of energy. So we either use ATP or the protomotive force, and we're going to take something and move it against the concentration gradient. And during that, so we'll take these molecules from the extracellular side, bring them intracellular, and at the end, they're all on the intracellular side. We moved them all together at one side of the membrane against the concentration gradient. So, for our concept check question, which of the following methods of transportation do not require cellular energy? Active transport, phagocytosis, penocytosis, exocytosis, or all of the above require cellular energy? You should check your textbook or rewind the PowerPoint to get the answer. The correct answer is E, all of the above. Obviously, active transport is going to require the use of cellular energy. By definition, it does. Pino and phagocytosis are two kinds of active transport that we talked about. They are two subcategories within endocytosis where we reform the cell membrane. Exocytosis is the opposite of endocytosis, and I did not talk about it specifically in this PowerPoint presentation, but exocytosis is when we reform the cell membrane to bring something out of the cell, to secrete, make a secretion outside of the cell. And these secretions that are going outside of the cell are generally speaking going to be against the concentration gradient. These can be molecules that are being secreted to the outside, extracellular side of the cell membrane. These could be secretions that are being secreted outside of the cell, maybe as an exotoxin. In any situation where you reform the cell membrane, whether it's endo or exocytosis, it's going to require cellular energy. Therefore, E, all of the above required cellular energy, is the correct answer. If you have any questions, please shoot me an email or post them on the discussion board. Happy studies!